Hi, everyone. Thanks for joining. I'm going to share my screen here in a second. So my name's Naomi. Nice to meet you all. I really appreciate the opportunity to present some of the work that we're doing at the University of Minnesota, and I'm very grateful to have this opportunity. So thank you so much. We're going to spend some time I'm going to spend some time talking. I want to leave plenty of time at the end for questions and discussion. All right, so let's get going. I'm going to give you a little bit of background about cancer, how cancer forms, and because that's important to how we prevent cancers. Talk about the journey so far in terms of chemoprevention specifically, and I'll talk about the definition of chemoprevention as well, and talk about some of the work that we're doing here, so the road ahead, and then loop back and wrap up for discussion and questions. So I think it's important when we're talking about preventing cancers or using chemoprevention to prevent cancers, it's important to remind ourselves what causes cancer. So there's factors that we know about, which are listed here, and several others that I probably forgot about. But it also turns out that many times we just don't know. So, which is of course a common theme in life. So what we can do is try to focus on the known factors. And I think it's also a good reminder to understand how cancers form. So DNA is a molecule or an entity in the middle of a cell, and it contains the instructions for making proteins. And you can think of proteins as the workhorse who is actually carrying out the job of the cell. And it's kind of straightforward in a way because it's only made, DNA is only made up of 4 bases. We call them adenine, thymine, guanine, and cytosine. We call them A, T, G, C for short. And basically what DNA is a code of A's, T's, G's, and C's in various combinations. And the idea is normally that DNA will transcribe itself into something called RNA, which is down here on the left lower corner. And RNA leaves the middle of the cell where the DNA is and goes out into the machinery that makes proteins in the kind of periphery of the cell, and then starts making helping the cell make proteins. And so then the proteins are made as you go along the bottom here. And it's these proteins that carry out the functions of the cells more or less. It's more complicated than that, but this is a basic idea of how it works. And how do cancers form is when there's mistakes or errors in the DNA code. So there's various types of mutations. And here on the top, for example, we have something called a missense mutation. And you can see the normal DNA code on the top. And on the bottom is the code with the error. So in the middle group of bases here, you can see that the G that was up here in the normal code was replaced by a T. And that creates an error. And so the DNA is not transcribed in the same way, and the proteins are not made in the same way. So the cell starts to do funny things. And likewise, we have things called frame shift mutations where a base gets inserted. So the A is inserted here, and then it kind of shifts the whole code over one, and so errors are made. And then sometimes bases are inserted that actually tell the DNA to just stop, so it doesn't do anything. On top of that, you can have chromosomes, which are bunches of DNA. You can think of them as bunches of DNA, and they can actually kind of break off and go to another chromosome and kind of do this flip-flop thing, and we call those rearrangements. And so all of these genetic errors can cause the normal machinery of a cell to go rogue, and so it's making proteins when it's not supposed to, and the cell starts to divide when it's not supposed to. Or likewise, you can actually turn genes that suppress cancer formation, turn those genes off, and so cancers can form. And that's important in terms of trying to figure out how cancers can be prevented. So there's some simple things that we can do. For example, we can reduce our exposure to sunlight, not get sunburns, wear sunscreen, and so on and so forth. So for infections, we can encourage vaccinations for HPV and hepatitis B, for example. We know those infections cause cancer. And we can treat infections such as H. pylori, which is a bacteria that can lead to the stomach cancer. And there's many other examples. We can encourage smoking cessation, and so on and so forth. So there are things that we can try to do to prevent cancer. And it's especially I want to point out the importance of screening. So screening is incredibly critical to prevent cancers and also identify cancers earlier on if they have already formed. And there's no question that cancer saves lives. And this includes things like prostate cancer screening with PSAs, breast cancer mammograms and breast cancer screening, of course, CT screening for lung cancer, and so on and so forth. So there are ways we can prevent cancer, and it's thought that maybe about a third to a half of cancer deaths can be attributed to things that we can control in our own lives. And this is specific to Western populations. So on the left here, you can see a pie chart where the relative contributions of each of these factors, such as diet, exercise, occupational exposures, primarily viruses or infections, et cetera, contribute to cancer death. You can see that actually tobacco is still a predominant factor. Tobacco use in the United States is declining, but it continues to increase in some other countries, for example, India, and it's still pretty prevalent in countries like China. And you can see actually that excess weight and obesity, which has not that long ago been identified as a risk factor for cancer development, might contribute a fair amount to the risk. So how does that really translate into our everyday lives? And that's really where the difficulty lies. It'd be really nice to say that we can do this, exactly this and this and this, and exercise exactly this much, or eat these kind of foods and really impact our risk of developing cancer. But it turns out that rather than, we're kind of a society that's used to saying we should exercise 30 minutes a day and go to the gym and whatnot. But I think rather than focusing on things that we can take or things that we can, or a very specific amount of exercise or the kind of exercise, I think just kind of changing the mindset in a way to just, and gradually too, to a healthier lifestyle in general. So, start to take really little steps in the beginning, especially, change just a little bit of your diet at a time rather than saying, I'm going to lose 10 pounds and that's really going to be the key to trying to reduce cancer risk and risk of other health issues. Just really changing that mindset and focusing on a healthier lifestyle in general can make a big difference. And on top of that, it's not just things that we can do for ourselves. It really takes a population-wide effort and infrastructure related issues, in addressing infrastructure-related issues, health inequity issues, to try to make an impact in preventing cancer. So I want to talk a little bit about the three steps where we can try to prevent cancer. And so there's primary prevention. So there's things that we can do to try to prevent a cancer from happening in the first place. And then there's secondary prevention, where we can try to limit the impact or progression of a disease process into a cancer. And then there's tertiary prevention, which is relevant, I think, to this group, which is trying to prevent a new cancer in someone who's had cancer. So not necessarily the same cancer, but even a different cancer, because we know that people who have had one cancer in their lifetimes, for the most part, are at higher risk for a second cancer. And so in the middle here, there's some suggestions about possible preventive interventions. And this is a paper that was written more for, like where is the field and what can researchers maybe focus on. But you can get an idea of some of the interventions that could be implemented at various stages of prevention. And the idea is that there's a lot more. There's just a lot more. And really the key to moving this field forward is understanding the biology of how cancers form and how the interventions that we're talking about can impact the process of cancer developing. There's a lot of challenges in this field, as in many other fields, but some unique challenges to this field of cancer prevention and chemoprevention. But the good thing is that people are paying a lot more attention, and that's really great, even in the last 10 years. So I'm very excited, and I think we'll see much more to come in the next 5 to 10 years, and even sooner than that. So we're going to switch gears a little bit and talk about cancer chemoprevention. So that's kind of what this talk is about. And one of the generally accepted definitions of that is the use of natural, synthetic, or biological chemical agents to reverse, suppress, or prevent carcinogenic progression to invasive cancer. So the idea is to use either a drug or a food or a compound or a supplement, for example, to try to prevent cancer from forming, and by specifically addressing the process by which a cancer forms. So, and there's a fairly, the history of cancer chemoprevention is not that long, it turns out. So back in the 2000s, people were really interested in understanding whether aspirin, for example, could prevent colon cancer. And there had been a lot of work along the last decade and plus. And we're still not at a point where people can say, yes, you should take aspirin to prevent colon cancer, but right now it's actually, the recommendation for aspirin is based on cardiovascular risk, but it is recommended, if you have a rationale to take aspirin from heart disease risk standpoint, it's thought to help contribute to preventing colon cancer. On the right side, we have examples of where chemoprevention kind of went bad, and this actually, the results of these sorts of studies and one other actually put a big damper on the field of chemoprevention for a while. But on the top here, we have a study where men were given finasteride, which is a medicine used for large prostate, and or a placebo to see if it could prevent prostate cancer from forming. And it turns out it kind of did, but the problem is that the men who got finasteride, who did get prostate cancer, got much more aggressive, on the whole, got much more aggressive forms of that prostate cancer. So on the left side column, this Gleason score tells you about the aggressiveness of a prostate cancer, and the low score is less aggressive, and a high score of 10, for example, is the most aggressive. And you can see compared to placebo, there were more men in the finasteride group who got Gleason score 8 and 9 cancers, for example. And so that certainly put a damper on that strategy. And then on the bottom, we have a pretty notoriously well-known study on beta carotene, so a micronutrient, a supplement, and trying to prevent the risk of lung cancer in smokers. And this is something called a Kaplan-Meier curve, and the idea is that the people who got beta carotene actually had a higher risk of developing lung cancer. So that was terrible. So because of these studies, the field of chemoprevention kind of came to a screeching halt for a little bit, and people understood that, number one, it was too expensive to look at whether a cancer formed or not, and then number two, we really had to understand how these things were working in order to determine any kind of benefit. We do have some examples of chemopreventive strategies at this point that we could potentially use. And so we talked about aspirin. Celecoxib is another anti-inflammatory drug that's been well studied. But, and especially for breast cancer, we have tamoxifen and raloxifene, which are two hormonal agents that can, that have been shown to mitigate the risk of developing invasive breast cancer in high-risk women. And then for cervical cancer, we have vaccines. So these are all examples of chemoprevention strategies, some of which are even in play now. But the field is really challenging. It's a really wide field. You can talk about trying to prevent a cancer. You can try to talk about trying to impact the causes of the cancer, and so the field is very wide. It's really difficult to identify the highest risk populations. So the easiest example here, and one that is near and dear to my heart, is that it turns out we all know that tobacco smoking is a risk factor for lung cancer, but it turns out that maybe about 17% of men, for example, who have smoked heavily will develop lung cancer. So that means there's a lot of men who smoke heavily who don't get lung cancer. And so we need to figure out how we can identify those people who are at the highest risk. And that's actually something that we're working on here at the university. It's also true that not all even precancerous lesions or things that we know could potentially turn into cancer eventually turn into cancer. A lot of them don't. And then not all precancerous lesions are the same. So you could have something like a cervical dysplasia, and and one person is going to turn into cancer and another person is not. And there's going to be differences on a genetic level and with other characteristics between those two lesions, and we haven't quite understood what those differences are. It's really difficult to define the endpoint in a study. So usually you do a study, you have a theory about what's going on, and then you say, okay, I expect to see these results. And we can't do studies where we're trying to understand whether the group that gets the intervention is going to have less cancer eventually than the group who doesn't. Those kind of studies take a very, very long time, and they're way too expensive. So even with tobacco smoke, we know that it can take 20, 40, even longer years to develop a lung cancer from smoking. And we can't follow, it's just too expensive and not practical to follow those people for that much time. So we have to identify surrogate endpoints, and then there's going to be debate about what's legitimate in terms of those endpoints. Because I think of the negative and even harmful chemoprevention studies in the past, it's been difficult to get funding to study chemoprevention strategies. That's changing. And then it's been difficult to get agents. So chemoprevention has largely focused, I would argue, on drugs or repurposing drugs to try to prevent cancer. Metformin for diabetes is a very, very easily identifiable and good example. But drug companies, for example, who often provide agents for studies, are not interested in really funding or cooperating with this kind of research. You know, they want to study interventions that help change the course of a disease. So that's been a barrier as well. Further down the road, it turns out that healthcare systems really aren't ready to widely implement chemoprevention strategies. For example, I mean, tamoxifen is kind of an easy example of that in terms of healthcare systems, as well as many other factors. But healthcare systems need to be on board as well, and that costs money, and they're not quite ready for that. There's a lot of health disparities, just like in cancer care and other areas. There's a lot of health disparities in terms of prevention, from screening, from access to agents that might be found to be beneficial, and etc. It's also the case where we can't be doing things that cause any toxicity, because ideally people are going to be doing interventions that prevent cancer for a long time. And they're also not going to have any symptoms, and they're going to be healthy for the most part. And so we can't be developing things that are going to cause side effects. And that's been a challenge. But on the other hand, there's lots of potential strategies to get around these barriers. And work and enthusiasm for the field has really picked up, I think, in the last 10 years. So I wanted to spend maybe just a couple minutes on diet and cancer prevention. So not quite chemoprevention, but diet, because actually what a big part of the work that we do at the university is focus on dietary interventions. So the guidelines say to eat a healthy diet, and the definition of what that means to the people who develop guidelines is here. And we've all heard these guidelines, I think, many, many times. It turns out that we're not, as a society, we're not that good at it. But you know, we can always keep on working on it. And I really think, going back to rather than focusing on specific interventions and saying this is going to reduce my cancer risk by this much, it's really about shifting the mindset into focusing on patterns of diet, rather than saying if I eat this many Brussels sprouts a day, it's going to affect my cancer risk. But it's really about patterns, and not really trying not to get focused necessarily on specific foods. And it's going to be more likely to be successful. You're going to be more likely to be able to stick to those changes over a longer period of time. So switching gears yet again, and starting to talk about what we do here at the University of Minnesota. So I don't know why I put his picture on the right side. It should probably be on the left side. But anyways, this is Steve Hecht. He's a world-renowned researcher in tobacco carcinogens, so cancer-causing agents in tobacco, and how those agents cause cancer, and also, importantly, how to measure people's exposure to these carcinogens. So he's a chemist by training, and I've told him many times I don't like chemistry. I don't quite understand it, and he just laughs. But he's a brilliant chemist, and has done a lot to advance the field of understanding people's exposures to various carcinogens. So carcinogens here on the left are cancer-causing agents, and it turns out that we can actually detoxify them and get rid of them. Either we pee them out, or the liver processes them and detoxifies them. But sometimes they get activated by mechanisms that happen within our bodies, and they they kind of latch onto DNA. And when that happens, it's called a DNA adduct. And those DNA adducts could then lead to DNA damage or mistakes in that code that we talked about a few slides ago. Sometimes our bodies can repair that DNA and it goes back to normal. But sometimes our bodies can't repair that DNA, and those DNA genetic errors persist, and then you get mutations in very specific genes that are known to, when they're knocked out, can lead to cancer. So here's some examples of well-known genes such as RAS or P53. And where Steve's work has focused on is identifying carcinogens. So taking tobacco smoke and actually identifying the chemical itself with really sophisticated techniques, and then developing methodologies to measure people's the metabolites of these carcinogens in the urine or the blood or in tissue. He's also worked really hard on understanding a little bit more about the metabolic activation process. So it turns out that individuals have different abilities to metabolically activate different carcinogens, and why would that be? So he's spent a lot of time studying that. So if, for example, if I were exposed to a certain amount of carcinogen and the person next to me was exposed to the same amount of carcinogen, we might have different abilities to activate that carcinogen or detoxify that carcinogen, and how does that impact our cancer risk or the risk of leading to DNA damage and stuff like that. So that's what he spent his career on. And leads to how we can try to use chemopreventive strategies to mitigate some of the processes that lead to cancer. So on the flip side, we have looking at carcinogenesis and how cancers form from carcinogens. And then on the left side, I should have probably had them flipped around, but on the left side, we have another very, very well-known famous person who was here at the University of Minnesota named Lee Wattenberg. And he was one of the instrumental people in starting the field of chemoprevention. And he and Steve worked pretty closely for many, many years. Dr. Wattenberg passed away, I think, maybe about 5 years ago or so. Not terribly long ago, maybe a little bit longer, but certainly after I became faculty. So I came into this being part of a long historic tradition of studying carcinogens and chemoprevention. And Dr. Wattenberg actually was very interested in studying vegetables and foods as a way to prevent carcinogen-induced cancers, and specifically something called cruciferous vegetables. So these are vegetables that include foods like cabbage and broccoli and some of the other ones that you see here, Brussels sprouts, and then on the right side, that's actually watercress. So Brussels sprouts and watercress and cabbage are things that we've studied here at the university. And why is this interesting? So if you look at population-wide studies, we call them epidemiologic studies. I know this is a little bit hard to look at, but if you take people who consume relatively little amounts of these vegetables, and how we get these histories is a bit problematic, but in any case, if you take people who don't eat very much of these vegetables and compare people who eat more and more and more, it turns out that the more you eat, if you eat more, then the risk of cancer seems to be lower. And OR is odds ratio. So for example, people in the second quartile have about a 40% reduced risk of developing cancer compared to the reference group here, and so on and so forth. And if you look at people who have smoked here on the bottom table, compared to never smokers, and risk of getting lung cancer, it turns out that amongst current smokers and former smokers, the more vegetables they ate, it seemed like the cancer risk decreased compared to people who ate very, very little of these vegetables. So what's in these vegetables that makes them special? So there's something called glucosinolates. And there's various ones. There's about 11 of them. And they get broken down into compounds called isothiocyanates. And there's various ones of those. And here at the university, we actually studied two specific ones, actually several. One called indole 3-carbinol, and another one called PEITC. And again, this is an abbreviation for a long chemical name. And why are these important? So it actually turns out that if we look at the urine of people who are exposed to carcinogens, we can show that by giving PEITC, the amount of carcinogens can decrease. And the number to look at here is this number. So the idea is that if in people who took the PEITC, and this was a study done about 5 or 6 years ago, the amount of benzene metabolite in the urine decreased by 95%. So that's pretty significant. And here's another carcinogen called crotonaldehyde, and in people who took the PEITC in general, it was reduced by 30%. So the idea is that these kind of glucosinolates can potentially impact a person's exposure to these carcinogens. And the work that I've been involved in initially started with studying indole-3-carbinol. And my colleagues in the lab showed that, for example, mice who got indole-3-carbinol and tobacco carcinogen had less lung tumors form compared to the mice who didn't get any indole-3-carbinol or its derivative, which is called diindole methane. So the mice who got indole-3-carbinol had less tumors per mouse compared to those who didn't get any. And so all that kind of work, you know, this is just a very superficial glance of what's the work that's been done. But how do we translate that into people? So one of the things about food-based chemoprevention versus taking supplements is the benefits, at least, is we're emphasizing a change in lifestyle rather than taking a pill to fix something. There's a lot more accessibility to trying to eat foods compared to taking a supplement. There's affordability issues, maybe not so much in the United States, but globally. On a global basis, it's not likely that people can afford to buy supplements as opposed to trying to eat different foods. And likewise, it's more sustainable if we focus on food-based chemoprevention. And the idea that really is the key here is that eating more is not necessarily better. So, and that's what we set out to figure out. And we did a study almost 10 years ago now where we gave people Brussels sprouts and cabbage. And Brussels sprouts that had these indole-3-carbinol. And on the bottom line here horizontally is the dose, you could say, of the Brussels sprouts. And so we knew exactly how much glucosinolate was in there. And then we measured the metabolite of the glucosinolate of the indole-3-carbinol in the urine called DIM. And if, as you know, over a certain amount of vegetables, the amount of DIM in the urine increases, so people are being exposed to more of the beneficial compound. But actually at higher, so you can call them doses of the vegetables, they actually didn't have any more DIM in the urine. So that suggests that there is actually a plateau, and eating more of the Brussels sprouts probably might not make a difference. So we did studies like that to establish that principle. And the idea is that supports the idea that more is not necessarily better. And this is on a daily basis. So these things get cleared very quickly, but you know, it's not sort of the thing where you can eat a pound of Brussels sprouts in a day and then not eat them for a week. It's more about, you know, eat some every day is the idea. And then we wanted to see if whether eating Brussels sprouts could affect the way that people process one of the carcinogens in the environment, as well as tobacco smoke. And so this is the first of 3 studies that I'll talk about that are ongoing. And in this study, we're actually giving people Brussels sprouts, and then we give them a very, very small, a very, very tiny dose of a compound that actually mimics carcinogen. So it's not carcinogenic. We can't do that. That's unethical. But we can trace this compound through people as they process it and detect it in the urine. And so we want to see if giving people Brussels sprouts can change the way that they process this chemical, this carcinogen mimic. So that study's ongoing, the recruitment's done, and we're analyzing the data. So I hope to have some data on this in the next 6 months or so. And that's important. That study is important because it would be kind of the first to show that actually eating foods as opposed to taking supplements could make a difference. We have a little bit of data so far. So these are let's see, about 9 people where we've analyzed the metabolite in the urine already. And in some of them, the amount of metabolite from before eating the Brussels sprouts compared to after, and this was a 2-week intervention, 5-day intervention, decreased fairly substantially. So again, not everybody, and we have to figure out why some people it decreased a lot and some people it didn't. But the idea is that it might be promising, and numbers are way too small to make any conclusions. We recruited about almost 40 people. So the second study that's ongoing now to look at food-based chemoprevention more or less is a study using watercress, and this is a study that's funded by the National Cancer Institute and that Steve Hecht runs. But based on the prior PEITC study, where he showed that giving PEITC as a pure compound could change the way that people process some of these carcinogens, again, focusing on food-based chemoprevention, he's studying watercress and looking to see if it can help people detoxify various environmental toxins, including carcinogens. So this is a study where some people will get watercress first and some people will get a placebo, and then we'll flip them around and see if there's any differences in the processing of these chemicals. The carcinogens / chemicals are ones that people are exposed to in everyday life, so we're not giving anybody anything to study that specifically. So that study just started literally about a month ago, and we'll probably take about another year or so to recruit. We have a phase 1 study that we completed looking at the effects of indole 3-carbinol and sylvanin on various biomarkers of inflammation and whatnot. So while we use supplements in this study, the idea is we want to translate into food-based intervention if the supplements look promising. And then lastly, not quite chemoprevention at this point, but I want to point out a study that we're going to get going, trying to study the effects of kava. So kava is a root that's commonly consumed in the South Pacific, and it turns out that people notice that despite their very heavy smoking rates there, men especially there seem to get less lung cancer than in other populations of heavy smokers, and they think that's potentially due to their use of kava. And my colleague who's now in Florida, who used to be at the University of Minnesota, has studied that extensively, both in just studying cells in a dish as well as in mice, and shown that actually indeed kava can have an effect on tobacco carcinogen-induced lung cancers. So we're planning a study where in actually people who have survived cancer, using kava and looking at its effect on anxiety and stress. So in the South Pacific, kava is actually used as a social beverage. They kind of grind up the root and make a beverage, and it's very relaxing. And so we want to see if kava can have an effect on anxiety and stress in cancer survivors. But in the same light, we'd actually like to eventually see whether kava could potentially have an impact on both carcinogen metabolism in people. We have some preliminary data to support that, as well as maybe even try to affect cancer risk, a secondary cancer risk in someone who's already survived cancer. So kind of a wide range of work that we're doing right now, as well as a wide range of work that's happened in the past. So where are we headed with all this? Well, ultimately, I and the others that I work with want to be able to recommend some dietary interventions. Again, not like saying, okay, eat this much of this vegetable every day, but generally speaking, some guidelines about interventions that can contribute to the prevention of cancer caused by environmental and tobacco carcinogens. Something that's doable and feasible, and especially we want to understand why it's beneficial. And we also want to see what the role of the immune system in cancer development is. That's not studied almost at all at this point, at least not very, very early on. And then we really want to do a better job trying to identify those who are at the highest risk of developing cancers from various carcinogens. So that's what we're doing here. And overall, this ultimately, of course, we want to reduce the burden of cancer. So I wanted to acknowledge all the people that have worked with me either currently or in the past. It's a very broad group of people, including our horticulturists who grow Brussels sprouts, for example, for us, and many others probably who aren't here